The Cardiovascular Research Center is just one of the sites on the UW campus where Petri dishes full of cells derived from fetal tissue are used for life-saving studies. Dr. Tim Camp and his assistants are currently looking at the proteins that cause heart arrhythmias by introducing them into fetal cells.
By doing an approach where we can study one channel protein at a time in the HEK293 cells, we can really zero in on some of these potential abnormalities and understand them and also test new therapies for them.
Those cells would be banned under the bill that Assembly Committee will vote on unless an amendment is introduced to exempt them. But the lawmakers who introduced the bill say such an amendment should not exempt the use of fresh fetal tissue, which isn't used in this study, but is being used for research in 14 other labs on campus. Associate Dean of Basic Research Richard Moss describes one study he calls vital.
It's the so-called humanized mouse.
That study involves injecting fresh fetal cells into mice that have no working immune systems.
So by actually developing a mouse that has a humanized immune system, we can actually test the efficacy of these cellular therapies in a system that won't automatically reject it.
They hope to develop new vaccines with that approach as well.
I don't think there's any scientist around who hasn't had that wow moment when things have taken a totally unexpected direction and breakthroughs are made. And that's happened with multiple projects involving fetal tissue and the cells derived from them.